Okay, so that's sheep skull. You can see two eyes here, nose, and the top half. And if we look closer, here is the ear canal of the sheep. And you can probably get size ideas from how I'm holding this. So that's the other side, and there's the ear canal. Okay, so that's the side we're talking about. Now, ear canal, right down the ear canal, you have tympanic membrane, then you reach the middle ear, and then the cochlea. So if we turn it around, as we can do here, we have this structure called otic bulla, and I'll show you the zoomed up version over there. But essentially, we have ear canal going down this way, and about the tip of where my forceps is where the cochlea is. So one thing to highlight here is that it's actually right underneath the skull, and it's only a few centimeters inwards from end of your ear canal for sheep. Typically maybe 3 to 4 centimeters in human, and that's the geometry information we kind of want for this drug delivery project, because it is very variable between the patient, and in some people it actively becomes narrowing, ossified and narrows human. So that's where we wear this. Now for poi, since you might be interested, so this is pink. So this is pink skull. And I've got goat over there too, and you can play when you come in. And pink, now pink, oh my God, they've got such a triangular cut off, like butcher's diagram. And huge difference is actually the ear canal here. So it's almost like goes in the vertical direction compared to sheep that was straight down. I was comparing the skull because we were trying to pick a large animal model perfect for device development model. If we are trying to develop an insertable device, we want kind of inner ear and skull geometry to be as similar to human without being a monkey. Looking at the crude comparison between pig and sheep, sheep was sort of round on the back, and the ear canal going that way, where pig is sort of vertical. I don't know about molecular features, but the size of cochlea is similar, but the skull and ear canal geometry is very, very different. And I wonder if I can just quickly find my goat. Sorry. So that's my goat, and this was kindly donated by a local hunter who had hunted a goat for their whatever industrial activity, because there's nowhere else we could get a goat. And the reason why I was also examining goat was because apparently it's easier to make genetically modified goat than sheep. Sheep is more established here in the department as a large animal model. It's used by many other groups. No one's heard of goat. To understanding they jump more. Looking at the crude comparison of cochlea, goat cochlea is a lot smaller, slightly smaller. I think 20-30% smaller than sheep from what I've seen. But obviously goat come as a different breed as well as a pit. Ear canal is here. And very similar to sheep, but you can see the huge horn. And when I saw this, I thought, oh my God, if I ever do research on goat, I'm going to get stabbed to death by goat. But because of the horn, you see the sheep I was showing is a hornless variation for females. This sort of mastoid or back of a head skull anatomy is quite different in goat, which I think it may also have a bit of an impact. So our preference is currently sheep, based on a crude assessment. And hopefully, boy, you can tell me more. Is that I've got a dissection microscope, and that's connected to this Zoom on this computer now. So it's just a live thing here. So I'm gonna just stick the cochlea underneath, and hopefully I can just turn it around for you. Okay. So Yep. Okay, so shown right here is a sheep cochlea, and I am as magnified out as possible, but it's with this dissection microscope is very good. And shown on the side here, this guy here. So this is a rat cochlea. So I might just focus on the rat first. And each of this block is about 2.5 millimeter. So I don't know if you can see much here. Keep on turning around. Let's see what I'm looking at. Oh, so this is the back end. Okay, so that's the back end of the cochlea. And if I turn it around, okay, so this corner here, that bit is where the cochlea is. So I might just annotate. So this here is a round window membrane, and I kind of have a cochlea tune here. Okay, so that's the rat. And with rat is one of the common model we use. Mice and rats both are commonly used. Rats slightly bigger, rats got slightly better hearing. Mice come with so many different strains, and they all have a different mutation sometimes that force them to develop an early hearing loss. So we need to be careful with the loading levels. And then comparison, this is sheep. So you can see it's huge, and you can take it from my word that the size is much more similar to human. Now you can immediately see so the bone. So what you're looking at is a bony external shell of the cochlea, and you've got not just circle around the round window here. Now oval window here. So this here is actually the stapes. Let's do that again. So this here is a stapes, the middle ear bone, like that. Here is a round window. So this is the right cochlea. And from stapes is connected to scala vestibuli component of the cochlea, and round window is scala tympani. So it goes, the cochlea turn should go kind of like that, like this way, underneath the bone. And the key point is that you can't see it because of the bone. Now, just as a tissue preparation sake, all these tissues are preserved. So they come from 4 to 6 years old New Zealand Romany sheep, which is the main breed used for research. White cute sheep, mainly females, because males can be quite aggressive, and we don't want to get injured working with animal models. And the tissue is kindly provided us from another group who routinely work with sheep for this particular sheep. But we are hoping to do some live sheep work of ourselves to do a hearing assessment on live sheep with Koyu as a main PhD candidate to contribute to that research. So the tissue is being extracted from a sheep head, and you saw the skull. So instead of using a dissection tool, this required us to buy saws and everything. So it's a bit of a hard work, then it's been immersion fixed in what we call paraformaldehyde-based fixative. So that's a very typical fixative. And what happens when we say we fix the tissue or we preserve the tissue is essentially you kill the tissue. And what fixative does is that it cross-links all the proteins together into sort of solid status. So the structure is preserved. Protein localization, if done well, is preserved. But tissue dies as a result. So what we have here is a dead tissue, dead cochlea with structure and a number of proteins fixed in place. Okay. I'll put here all drawings. So that's the original tissue. And I might just bring up human tissue. in comparison with the hell is it? Okay, so that's a sheep cochlea. Now I don't have a full human cochlea, but I've got a half a human cochlea. And now, sorry. So this is a human cochlea. It's been processed in a pretty much analogous way, but the bone's been trimmed quite extensively. So I might just flip it around. Okay, so I think you can see the tunes. So this part has been sectioned, and you can hopefully see the cochlea tune like that. So this is a human cochlea. Human cochlea comes from post-mortem tissues, and the University of Auckland has a body donation program, and some of that's used to train doctors for dissections. But some of the tissues also available for research purposes, and we recently got an ethics to work with these tissues so we can get the ear and we can do research. So it's all preserved as well. And okay, so you can probably hear what's going on in the background. Okay, so just side by side, on the right is a sheep cochlea, and again we've got this sort of turn situation here, and we've got this turn situation here. So I guess rough size wise, you can see that it's quite similar. Okay, so that's the standard preparation. Now for micro CT, what happens is that essentially what's close to what we've got, so like human tissue sample here, it gets scanned for micro CT. And for micro CT's purposes, to get a good image, it's all about penetration and how to maximize the penetration of X-ray, but also how to change the absorption of X-ray by the different location of the cochlea. So first thing that we typically do is if we, for example, want to look at the cochlea, we trim the bone away as much as possible. And bone's been trimmed away here because bone's been softened using this solution called EDTA. Now that's a buffer which chelates calcium. So what happens is that calcium over time dissolves into the buffer, get chelated by EDTA. So more calcium just keeps on dissolving out. And the whole process takes 8 weeks for sheep cochlea, and somewhere between 3 to 4 months for human cochlea, with constant changing solution and trimming of the bone. So it's a long time effort. Because of the size and the thickness of the bone, but in the end, it's soft enough that we can section. If we want to take a tissue section, and we can trim away bits of bone by bits of bone as needed. So that's sort of. And if we want to look at the cochlea, we'll trim away, centralizing the cochlea. If we want to look at the round window, we'll trim it right down to round window. So tissue preparation is a lot of hard work. And from here, for micro CT, we get something like this. And so this is a sample that's probably generated the data that I've just shown you as example for micro CT, and we have something that Kahi's got. Now it's completely black now because it's been stained with a molecule called osmium, and osmium is something that enhances the contrast of X-ray. So otherwise, if you don't stain it, not much X-ray actually gets bounced around by the bone and the soft tissue. So soft tissue becomes almost invisible. And just like your chest X-ray, you primarily see the bone and you don't see much of the soft tissue. It's hard. So to help us visualize the soft tissue inside the cochlea, the typical tissue cochlea scan is done with this osmium staining. And then gets scanned. So this is a typical sample that we've scanned to generate the cochlea data. Now, cochlea data might be something that you, Senorita, and Brace may also be working on as well, to get some of the features from human cochlea and sheep cochlea, because we're going to use that information to build a model system to do a few experiments around drug delivery. So this would be also, again, similar material that's generated data you could be working on. So this is a human ear bone, similar to what I had for sheep, trimmed right down so that we've got an ear canal and in the cochlea. But just to sort of now sticking this under the microscope, this is an ear canal here. And if I hopefully go up and down, you see there? So that's your round window. So this is, I might just rotate it around. So this is a ear canal, and maybe that's better. And if I tilt it this way, this is where the round window is of the cochlea. So here's a broad ear canal. I think that's maybe this way. The cochlea right under the nose. And hopefully you'll see the micro CT data that the whole thing is being scanned. So that's again another type of image that you might see. Now the sheep cochlea can be dissected further, so I'm just going to show a few. Okay, so this is a sheep cochlea, which was, you guys were before looking in this orientation. Yeah, so you were looking at before in this orientation, and then it's been cut open. So now it's gone soft enough. We can do this. Okay, so if I focus on that and maybe change the light a bit. And I hope you can see a bit of a contrast here, but here's the oval window. So this is a stapes, like an end of stapes connected to the cochlea. And you see this cochlea turn here. And if you look really, really closely, you might also see this radiating nerve fibers coming from modiolus towards the outer end of the cochlea. So again, I think from tissue biologist's point of view, cochlea is a beautiful structure. It's so complicated and very hard to dissect. I think it's a biological beauty. What was this? Can't remember what this was. This is another. Okay, so this is another example. And so this is now sort of cochlea bits being cut out. Okay, there we go. Okay. So again, this is a sheep cochlea. So now half the bone's been peeled off with patient dissection. And you can see this nerve, thick band of nerve in the modiolus, and there's a cochlea turn. Now you see quite a stretch of pigmented cells. So this is the area of the spiral ligament that contains a lot of vasculatures and fibrocytes and supports the physiology inside the cochlea. It's actually quite funny how pigmented it is and why you need a pigment inside ear, but that's something we see all the time. And to be honest, for microscopy, it's actually a little bit of a pain. And over here, this time is just another cross-sectional view, I guess, of a human cochlea. So clear. So now we've got a mushroom shape, and we've got a bit of a like different chambers visible. So this is modiolus coming in. There's an apical turn, there's a basal turn, and we've got the scala tympani, scala media, and different cross-sectional views. Again, if we cut in half, you can see that. Now, then part of the cochlea can be dissected out. Where is it? There you go. There you go. So over here, you see this thing here. So this is part of a cochlea that's been dissected out. So originally it was turned like that, and the strips have been cut off. And this henna is a type of data that you see for some of the radio images. And I might just zoom in a bit more. So this is what we call whole mount preparation, because it's not a tissue section, and it's mounted as like a whole tissue. And if I zoom in a bit, mounting is everything. Now you see quite a slightly different contrast here along the way. So this over here is a nerve fiber. So over here is the center of the cochlea. This was the end of a turn, and it normally goes around. This semi-transparent bit in the middle is where the sensory cells normally reside. So your organ of porta and basilar membrane of the cochlea is even here. And the vasculature of the spiral ligament lives around here. And organ of porta here. The nerve bundles for auditory nerve, the cell bodies are located on this side and they radiate out to reach here cells. So that's what you're seeing as a bit of a fibrous structure here. And vasculature coming from modiolar artery side also branches out in this sort of radial format. So this is where some of the radial vasculature image comes from, henna, for your henna. So that's a different type of preparation, and I'll finish off with showing an example of a tissue section. Now for microscopy, we needed things to be thin because microscope has such a narrow range of section focus, and we kind of need various things to penetrate as well, like dyes to penetrate. So it's often a best idea to section. And thinner it is, crispier image you can get. Dye works well, but harder it is to keep the morphology. So there's a lot of optimization that happens in the lab status. So this is a cross section of a sheep cochlea, and here is a round window. And you see the bone here. And this is the inside the cochlea. And this is outside of the cochlea. So that's one cross section. But as you can see, if I shake it, it's a thin floating section. So we tend to label it this way and mount it on the slide at the very end. And what you're going to see is completely going to be dependent on how you section the tissue and the angle you section the tissue, because it's such a complicated architecture. So with anyone's project, with any data analysis, first thing that you're going to have to familiarize is to have the cochlear geometry with a turn, round window, oval window in your head. So as you look at the any cross-sectional view, either from micro CT or CT or MRI or sections, you need to then visualize in your head and work out which angle you're looking at, which area you're looking at, what scale you're looking at. So it's a bit of a challenge. I think it's quite fun. So that's another cross-section here. So I might just point out to this one here. And there's another section here, so that's a bit confusing. But now you see a cochlear turn here. So this is the modiolus, outer shell of the cochlea. There's another one that's quite a typical of the the diagram. So you see this chamber thing here on this side. Clear and draw it again. So you see modiolus, and this is the external side of the turn. So this is the cross section of the turn. You see the white bit with a nerve fiber coming out to this way. Basilar membrane and organ of porta here. This is the spiral ligament or the periphery. This is the center. And we have this cochlear cross-sectional chamber here, one here, and then we have another one here. And so another spiral ligament here, another nerve coming out here, an organ of porta and basilar membrane here. So now this is taken from the same cochlea, but it looks totally different because it's taken from a different location. So again, and what you can see is completely different. And the difficulty often is having a comparative location to compare from one data set to another. If we want to see any changes with pathology or individual variation, we have to first find a comparative plane to compare out of this 3D structure, because depending on the angle and location you're looking at, what you can see and visualize is completely different. So that's a tissue section, and it's floating because it's this one is 30 micron thin. For something that requires thinner ones with more resolution, for this type of sections, we've first frozen the sample in section, then we can go down to about 10 microns. If we want to do an electron microscopy, we have to go to nanometer 6 sections. So that's the tissue section, and that's about it. I might just finish with one exciting note, because I love visualization, and hopefully throughout this demonstration it will show that I love my work. So shown here is the same cochlea, sheep cochlea, as I've shown before, but now I'm trying out this new thing for tissue clearing. So the whole cochlea, including the bone, so we've still got the bone, it's been made transparent using a tissue treatment. And the amazing thing about it is that, so it's the same thing as what you were seeing before, this thing, but it's been immersed in a solution sequentially for a whole week to make them transparent. And it doesn't look that clear there, but if I get it all in here, see how clear it is? Yeah. And see, you can see the turns now. See there? So you can see the beautiful pigmentation and the turn coming around this way, and bone is still there. So without dissecting, now what I need to do is bleach this pigment, because it's actually in the way of fluorescent imaging. They absorb too much light. And find a way to label certain structures in this transparent preparation. And we can image the whole thing if we can find the right imaging setup. So instead of taking tissue section or microscope or micro CT, we can hopefully get a similar type of data set. And I'm talking to someone about that. So there's quite a bit of things that we can do with in this space. Okay. So